All right, folks, here we are, our destination for today's lab tour. This is the Mary Ellen Jones building. This building was originally built in 1978, thereabouts, and it was dedicated to a medical school professor and department chair, Mary Ellen Jones. Now, she was the first woman at the entire university to occupy an endowed chair position, which, we can learn what that means later. And she was also the first woman to hold a department chair position at the entire university. She was also a biochemist like yours truly. She did amazing science. She was an amazing leader. And it's no wonder that they decided to name this building after this incredible leader and scientist in Mary Ellen Jones. And so we're going to go in today and meet with one of the medical school professors. He's actually co-director of the MD PhD program, Dr. Mohanish Deshmukh. And so he'll be down momentarily to just let us in and take us around the tour of his lab.
Warm welcome to all of you at UNC. My name is Mohanish Deshmukh. I'm a professor here in the cell biology department and the neuroscience center in our med school. I've actually been here at UNC for now 20 years. Hard to believe that. So I started here when I was 5 years old. And time flies. I have loved it over here. We do cool research on neuroscience that I'm going to tell you a little bit more about. But in addition to the research, I'm also the co-director of the MD PhD program. And so I'd love to talk to any of you about MD PhD programs if you'd like to get some information. Yeah, that sounds good. Yeah. Let's go inside. And
all right, let's go inside and take a tour of Dr. Deshmukh's lab.
Wonderful. Okay. This is actually a brand new, well, it's not a brand new building, but it's a renovated building. It's called the Mary Ellen Jones building. I'm sure you talked about that. This building actually existed more than 30 years ago, but it has been really made to look brand new. The shell, outside shell is the same, but the inside, everything is new. Just opened about 6 months ago. And so we were fortunate to get a lab space over here. The entire neuroscience center is over here. We're going to go up to the 5th floor. This is 3rd floor, so only 2 stairs. And we'll go this way. The building actually has biomedical engineering people and pediatrics research. So it's a mixed space with labs, different kind of departments over here. Neuroscience Center occupies 3 floors. Over here. All right, I'm glad my lab's on the 10th floor.
Me too.
Okay. Normally we would have a bunch of, this actually is the microscopy board. It's got really sophisticated confocal high-end microscopes for doing research. And so our lab actually starts from here. That's Healy, our grad student over here. And then we have space over here. That's Sahil, the lab manager. And so we have about this much space. Then we have amazing colleague, Dr. Ava Anton, on that side of the lab. So it's basically Ava's lab, our lab, and then a new faculty is going to come to that space over here.
So you told us a little bit about your background outside, but tell us how it is that you ended up at UNC and why you love it here so much.
Yeah. In our sort of career path after you do your PhD and you do your postdoctoral work, then if you're interested in academia, you look for a job over there. So I knew, of course, about UNC before, but it wasn't the case that there was anything other than the fact that we had a job posting for an assistant professor at that time. The Neuroscience Center was actually a new center being established at UNC in 2000. There was a director who was recruited, and I did happen to know the director's name is Dr. Bill Snyder. And so when he knew that I was in the job market, and asked me to come here, I did visit a few other places for faculty jobs. I just love the people over here. Chapel Hill is a beautiful area. And so even though some other places were giving me more space and a little bit more money, at that time I decided that UNC was really an amazing place for me to be here. I think if you asked me in 2000 whether how long I would stay here, you know, I even right, yeah, I mean, it's hard to really imagine that you'd be in one place for as long a time. This is the longest we've ever stayed in any place. But you know, there are just fantastic colleagues here. It's a great university, wonderful students. And so to me, there hasn't been any compelling reason to even leave this place. I mean, I could easily see being here for the rest of the time. You need good students, you need good research facilities, and you need good colleagues. And this place really has all three. And I get to work with people like you, Freeman, you know, and students from the CSS programs and the MD PhD students. So, and you know, just look around. This is, why would you go anywhere else? This is not what my office looked like, you know, first 15 years, but it's just fabulous, actually. So.
That's quite a view you got there.
Yeah. So.
And how long have you been co-director of the MD PhD program?
Yeah, so that has been for the last 3 years.
3 years. Okay.
I've been associate director 3 years before that, and I've been associated with the program pretty much since the beginning of here, in the admissions committee and things. We've had 3 wonderful MD PhD students who've done their PhD in our lab, and so it's really a great opportunity to recruit some of the best minds, people who are motivated to be a physician, scientist, and we actually recruit really outstanding underrepresented minority students to this. Of course, we want to do a lot better, but we are among the nation's highest for recruiting underrepresented minority MD PhD students over here. So for me, it's just an amazing opportunity to work with fantastic students and colleagues. And see them succeed and do really well. Particularly in this time, more than any other, that's what I was telling the students a couple of months ago, at this time more than any other, you know, the need to be physician scientist is no greater than it's ever been. This is really an amazing career for people who are considering it.
Absolutely. So now we're about to go take a closer look at your research. Would you mind telling us just briefly what your research is about and, you know, why your research is so important?
Yeah. So fundamentally, what my lab studies is how cells survive and how they die. That pretty much covers the entire thing of biology. It's simple, right? We want to study how cells survive and how they die. So, and everything in between. Now, I mean, what we really study is how cells think about cell survival and cell death. And you guys may be familiar with the fact that there is a dedicated program for killing cells. It's called apoptosis. And so that's really at the core, that's what we study. How does a cell decide whether it wants to continue living or fix the problems it may face, or actually activate the cell death pathway, the apoptotic pathway, and kill itself? And so, and what's amazing about it is that different cells think about this differently. And that's really what my lab is most interested in doing. And I'll give you 2 examples. On one end of the spectrum are cells such as neurons that really don't want to die, that are really built to survive. And when you think about it, your neurons that you're born with, for the most part, actually are with you for the entire life. So the cell death pathway has to be tweaked differently to make sure that these neurons can actually live for a long time. And so we've discovered amazing mechanisms that neurons have evolved and engaged to maintain their survival long term. And this is an important point to make, because, you know, people rightly so talk about neurons dying in all these various diseases. That's where the most focus is. How do neurons die in Alzheimer's, Parkinson's, or Huntington's disease, we've actually flipped the question, and we are more interested in asking, how do the healthy neurons survive pretty much in the most of us for the most of our lifetime? And shockingly, there are not that many people who are asking that question. Most people are focused on why they degenerate. I'm not saying neurons don't degenerate, but we want to discover what are the mechanisms that enable a healthy neuron to live for 60, 70 years of their life. There's not a single other cell in our body that can do as amazing a job. So we study and celebrate the survival of neurons rather than focusing on the death of neurons. So that's one extreme, which is neurons are very resistant. On the other extreme are the embryonic stem cells. So we study human embryonic stem cells, and I'll show you some of these cells hopefully. And these cells are incredibly sensitive to things like DNA damage. Why? Because if you're an embryonic stem cell and you're giving rise to the entire embryo and the human body, then you want to make sure that the DNA is perfectly intact, because any mutations that carry over at that early stage will spread to all the cells in your body. And so these cells, the human embryonic stem cells, we've not seen any other cell that dies so quickly in response to DNA damage as human embryonic stem cells. Mm-hmm. So we're fascinated about the fact that how do different cells think about survival and death? And we study human embryonic stem cells, how they are so sensitive. We study neurons, how they are so resistant, and how one goes from sensitive to resistant to better. That's really the core of what my lab studies.
Awesome. Now I'm going to ask you maybe a controversial political question because you brought up embryonic stem cells, right?
Yeah.
Human embryonic stem cells, yes. And so how has, various changes in political climates affected your research over the years?
Yeah.
Has it?
Yeah. So, this used to be a lot more controversial topic maybe 15 years ago, and we've been working with human embryonic stem cells for more than 10 years. What happened about 10 years ago is that the Bush administration actually identified a set of human embryonic stem cell lines that they said researchers can work on these lines. So most people are not looking for new sources of human embryonic stem cells. That's where the controversy is. The ones that are already approved by the government, by NIH, are the ones that we work on. We just work on one of them. But it's not an easy point to make for people who are not that knowledgeable. They think that you're harvesting the human embryonic stem cells. Right. That's simply not the case. These are cells that are around. They're fantastic cells, and that's all we need for doing our research. The other thing that's happened is that the promise of really usefulness of these cells for disease has really become very apparent to people. And so the benefits of doing this research vastly outweigh the negligible costs, particularly if you're working on cells that are already there. Mm-hmm. The other thing you may well be aware of is that the technology now allows people to get your skin cells and convert them into what's called induced pluripotent cells. Mm-hmm. They're called iPS cells. And then the iPS cells are basically like embryonic stem cells. They can be made to grow into a neuron or a skeletal muscle cell. And so you can actually get these cells from your skin of the patient, convert them to iPS cells. So it's really, I think it's become a not as controversial point anymore.
Good.
For us, scientifically, they're just amazing cells because they are cells that can exist in a undifferentiated dividing cell, and then a few weeks later they can become a neuron. And so it's just incredibly powerful to see how things change from one state to another. Mm-hmm. And so we do those kind of experiments.
Awesome. Now, before we go and look at how you and your group try to answer some of these pressing questions that you have, what has been your most surprising result that you've seen in your career?
I think it's the same point about neurons having multiple mechanisms to stay alive. The reason I hesitate to say that it's surprising because if we had just thought about it, it would have made sense that these cells actually live for a long time and have great mechanisms to stay alive. But Thomas, I'm telling you, you can go to the neuroscience meeting today, a room full of hundreds of neuroscientists, and if I tell them that neurons have amazing mechanisms to stay alive, they will think I'm a fool because they're so hung up on the fact that neurons degenerate that I've been so surprised at the reaction I get from my colleagues when I tell them neurons have amazing mechanisms to life. And then it's like, yeah, actually, that does make sense. It absolutely makes sense. I'm not saying neurons don't die, but even, let's say, Alzheimer's disease, it's only after 70 years or so that the neurons will start degenerating. So why ignore the first 70 years of the fact that these single neurons are actually surviving in an amazing way? I think it's one of the vastly underappreciated aspects of the biology of neurons. And I'll add one more thing to it, which is that you appreciate how good a job neurons do at surviving only when you compare neurons with other cells. And most neuroscientists really are not that interested in other cells. So they work primarily with neurons. And they take neurons for granted. They're like, okay, these are cells that are wired heavily. They do all these important things. And they sort of completely miss the point that if you just compare the neurons with other cells, which is what we do, then you realize, oh my God, these neurons are amazing in every possible way, including survival. So that's the biggest surprise. But if you had thought about it, it shouldn't have been a surprise to us.
Yeah, but always obvious in hindsight, right?
Yeah. Yeah.
All right. So shall we go?
Yeah.
See what's going on in the lab?
Yeah, absolutely. So basically, just coming back to the lab, our lab is primarily a sort of a molecular cell biology lab. A lot of the work we do is on neurons, and so for neurons, we have to isolate them from animals. So we work with mice. Whatever neurons we need for our experiments, we have to dissect and isolate from neurons. We don't have a dissection going on today, but that's what we pretty much do on a regular basis. And it's amazing that these neurons that you get from mice, you dissociate them, put them in a dish, and they absolutely will do everything that a neuron does in a dish. They will grow, they will give out these beautiful axons. And then what we can do is induce the cell death pathway in these neurons with some tricks, and then ask how does that happen. And then we can manipulate the genes and see what happens if you modify genes in the cell death pathway. Do neurons live, die faster? And that's how we discover how neurons make a decision to survive or die. Broadly, there are 4 things my lab does. I mean, one is looking at cell survival and death in neurons. The other is looking at cell survival and death in embryonic stem cells, like I said. Embryonic stem cells also during the course of differentiation. The third thing we do is back in neurons, we are very interested in knowing how does a neuron regulate its cell death machinery differently in a cell body versus the axons. You guys may know neurons in the body have this amazing, beautiful structure of a cell body, the nucleus, and then very long axons, right? So there could be axons going from all the way here to the tip of your feet. And it turns out that the neurons can selectively degenerate their axons differently from the cell body. And so we're very interested in knowing how can a single neuron spatially restrict the cell death components to one part of a neuron, one axon branch. In some ways, this is exactly what we do every night, actually, when we go to sleep, when we dismantle some synapses that we don't need to keep for the next day. We see so many things every day. We don't need to remember every one of them. I don't need to remember that you were wearing a blue shirt. But now that I'm saying it, I will remember tomorrow. But I didn't need to remember that, in which case the synapse that registers that information is actually destroyed. But if you came and just slapped me in my face, I need to remember. I need to remember that. So you strengthen and weaken synapses over there, and that's also by this same kind of cell death machinery that selectively is activated. So we use these microfluidic chambers to study that, and I'll show you what those look like. And the fourth thing is we do a lot of in vivo experiments in mouse models, and we have mouse models of Alzheimer's disease, and we have mouse models of autism. And I don't want to go into all the details, but those are amazing models where we look for new therapeutic approaches to prevent Alzheimer's disease, and also in the context of autism, what might be happening there. So it's a whole spectrum of things, but survival and death is what we are most interested in.
Nice.
So I'm going to show you some. We'll see some neurons. Hopefully, we'll see some embryonic stem cells too. and we'll show you some of these microfluidic chambers.
Okay.
So this is Matt Gedden. Matt's a postdoctoral fellow in the lab, and I requested Matt to see if he can see some movies. We've got a high school student who actually made facials for us,
Oh, nice.
and they're pretty amazing actually.
Whoa. Let's see. Let's take a good look at these things.
Yeah, if you can get like a UNC
Look at
logo in the
that.
back.
Look at that. Thank you. All right, we're all face shielded up.
Yeah.
Ready to go look at some neurons.
So I just want to show you these images. These are what the neurons look like in these microfluidic chambers. Okay? And there are three compartments over here, and the neurons actually are plated in the middle compartment. These little circular things you can see, those are actually the cell bodies of a neuron. The nucleus is over there. And these spectacular axons can extend from individual neurons, and then we have them going through these micro grooves into either compartment. So they look absolutely beautiful. But they're all coming from these individual neurons, the axons in either compartment. The reason we do this in the microfluidic chamber is because it allows us to expose the axon tips to stimulus that is independent of what we do in the cell body. And normally you wouldn't be able to do that because a neuron would be in a dish and everything is exposed at the same time. But now we can expose this compartment and this compartment to different stimulus. And so it's a really cool way of spatially isolating the stimulus for neurons. I should mention, this also looks beautiful. It's actually won several science and art competitions. Mm-hmm. This very image, which was taken by Matt and Selena, another grad student, was actually featured in the Ackland Art Museum last year. And it's gotten some other awards. So we're thrilled that some of the things we do not only are scientifically important, but also look visually stunning. For that. We're thinking about printing T-shirts about this.
Yes, absolutely. Was this when they had the Kahali Ramon?
Yeah, exactly. Yeah,
I think I saw this.
Yeah, we actually they had a competition, and three were selected to be displayed over there.
Yeah. So, quick question.
Yeah.
So you said these big circular parts are the cell bodies.
Yeah.
Have any of those escaped out into this area?
No, it's not possible because the micro grooves are too tiny. I think they're about how thick are the micro grooves?
These are about 15 microns.
13 micron, and these are 30 micron or so.
Yeah, they just don't go through the grooves during the waiting process. We can stain for them and we don't see anything. So it's a very robust system.
And when I say they're fluidically isolated, that means even though these neurons are connected, they go through these grooves. If you put a red dye, for hypothetical case in this compartment, it will not go into this compartment.
Oh, interesting.
So that way you can expose this differently to this part. And that's the best part about these chambers. And by the way, this is a great example of something we were able to do because of the fact that we had access to wonderful people in the chemistry department, and it's a microfabrication laboratory called Channel. We approached them with some designs. We had help with one of our faculty colleagues, and these chambers were made right here. And so it's just a good example of how different departments can work together to just help each other out. That's
very cool.
Okay, so let's see if Matt can show us something else. So these, by the way, these are the tissue culture hoods that we use for working on cells. You have to work in sterile conditions, and so when we dissect them out, we typically work on these biosafety cabinets over here, and then put them in these chambers. Yeah.
I was going to see if I have some brain slices somewhere.
You have brain slices over there. Yeah, let's just stop over there for a minute. Yeah. So here's a great example of a mouse brain section that's been stained with different antibodies. We don't have to go into details, but for example, the kind of things we do look for here is if you compare a normal wild-type mice with a mouse that is deleted for a particular gene. Let's say, for example, that gene is linked with schizophrenia. We want to know in a mice that doesn't have this gene, how does the brain structure change? And so we look at the section, we stain it with different antibodies. This, for example, is the hippocampus right here. The cortex are these layers over here. And so we can look for malformations or changes in expressions of proteins by doing this kind of a staining. There are multiple colors used here because that allows us to look at 3 or 4 different staining patterns for 4 different proteins at the same time. And so that's very cool.
Videos?
Yeah, videos would be great. Matt has taken some movies of neurons, which always, this should give you some idea of how beautiful these neurons are and what they do in culture.
You want to see them being plated first?
Yeah. Yeah, so this is from the start of the movie, and we can play that again. They were plated, and you saw they start off with tiny circles, but within hours, they give out these axons, and within a day or two, the entire dish becomes, completely sort of full of these beautiful axons. So starting off with individual circular cell bodies, and then neurons in a dish will do what neurons in vivo in animals will do here. So this is a movie of neurons that are exposed to a DNA-damaging drug. And you can see they start off perfectly normal, but after a few hours of exposure, you can see some of these individual neurons just dying and popping off. And while it seems to occur fast, this actually takes about 24 to 48 hours. And we're most interested in knowing not what happens at the very end stage when they pop off, but actually throughout when the neurons make a decision on whether it should survive or die. In this case, I made a big deal about neurons surviving. In this case, we're exposing them continuously for a very high dose of DNA damage. And neurons are doing everything to stay alive, but eventually they will die. You can also see these non-neuronal cells sometimes moving around, and they're basically looking around to see whether they can phagocytose the dying neurons. It's probably better seen in another video. You can see another non-neuronal cells. These are cells that are basically just looking to make sure that everything's survived. If it's not, they will come in and just eat away the degenerating neurons. So please.
So these are actually images from like the triple chamber setup, where the axons are coming through here and the cell bodies are over there. And what we've done is we've actually tagged the mitochondria with fluorescent proteins, and all of these little dots here are individual mitochondria, and we're watching them in real time. This is maybe a 5-minute video, and you can see the mitochondria just shooting along these fine axons, just moving at incredible speeds. And during cell death, mitochondria are like a critical component of how that occurs, and also in a lot of neurological diseases. So being able to look at the mitochondria specifically and not have to worry about affecting the cell body for our experiments is a really interesting and cool way to do ask a lot of questions for apoptosis research and axon degeneration.
One of the things I'll point out is it's amazing that not every single mitochondrion move. There are a subset of mitochondria that move very rapidly, and others that just stay. And we have no idea at this time why some move and why some don't. But there's something really different about individual mitochondria. And when they move ridiculously fast. Mm-hmm. This is in real time, right?
Yeah, this is like a 5-minute video, but they're moving very long distances. Like this is maybe a millimeter. So they're moving incredible speeds for considering how small they are.
Yeah. Los Angeles freeway over there.
I've got non-neuronal cells wavelength, but that's it. There's another one. That's way there.
That's beautiful too, yeah. What are those?
So that is a non-neuronal Schwann cell. Non-neuronal Schwann cell, which is a support cell that normally wraps itself around the neuronal mitochondria, or around the neuronal axons, that helps myelinate the sheath, so that the signals can transfer from long nerves down throughout the body without the signal decaying. So we have here is one of them in culture, where it's interacting with the neurons that we've grown in culture. And this is a video of the mitochondria of this support cell in culture moving naturally and just kind of doing native behavior. But it's so much more dynamic than you think when you look at a cell. You just look at a cell, a lot of times the image is static. This, I think, is a 1 minute video. So there's just a lot of dynamic processes going on in these cells that you just can't see unless you use cool techniques like this fluorescent microscopy in lab cells.
It also highlights some of the differences in the length of the mitochondria and the movement, because here the mitochondria are actually much more elongated. If you look at your biology textbooks, there's a static image of a mitochondria. It's probably the same image of mitochondria that everyone's seen since 1960s. But mitochondria are highly dynamic. They can fuse and form these elongations, and they can fizz and separate. And the ones that dart across the neurons are actually the shorter ones, because it makes sense for it. And actually there are diseases, some of the neurogenic diseases occur when these neurons cannot fizz, cannot separate, because then they can't send energy all the way to the tips of their axons.
And I love this video, especially because this series of mitochondria here, it's those two start off as two mitochondria at the beginning of the video when it starts again, and then they fuse into a single larger mitochondria. The kind of behavior that a lot of times we discuss hypothetically, but being able to actually see it occur in real time.
Within a minute.
Within a minute. It's wildly dynamic and wildly fast, and you would have no idea if you look before and after.
Yeah.
It's very cool.
That sounds cool. Guys, this is Keely. Keely's a grad student at UNC, actually just joined our lab. Welcome. To have her in the lab. She just finished first year in the neurobiology curriculum. And she's going to show you some pictures of neurons that are stained with what Keely does best.
Okay. So these are SCGs, which you guys may have already talked about. But they are neurons that are stained with tubulin in red, which is just a supporting structure within the neuron. And they're also stained with hoaxed, which marks the nucleus of the cell, and cleaved caspase 3, which you can see in green, which is a marker that the cell is undergoing apoptosis.
Yeah, so that's really cool. So the cleaved caspase 3 staining marks the cell that is about to die very quickly.
Yes, these green ones are the ones that are dying.
Yeah.
So I can show you also a video of this. So
Oh, we saw that.
Oh, you saw this already. Did you see them dying?
No, actually we didn't.
Okay, so this is what the cells look like as they're plated. So they grow within the dish, and then this is what they look like when they start dying. So as you can see down here, the cells almost look like they're kind of popping, and then they're getting degraded.
Yeah, and this death mechanism is slightly different from what we saw earlier. This is in response to deprivation of a survival factor. And you can see after the axons have degenerated how these non-neuronal cells come and just eat them up much more clearly over here.
So those non-neuronal cells were the ones that were kind of still moving around. Go back to that. So here's it. Oops. There's a good non-neuronal cell down here.
Okay. Wonderful. So
Right.
last time I'm going to ask you, what are you doing today?
Today, I am doing an RNA library prep for something called RNA-seq. And basically how that works is you take tissue samples and you put them through a process of a whole bunch of different reagents and chemicals that work to isolate the RNA, which is the untranslated protein. And from that RNA, then you can compare the proteins that would be made in a variety of different tissue samples to see if there's anything interesting and different between them.
Very cool. Thank you so much, Keely.
Yeah.
Last thing I want to show is, Sally, can you show us some human embryonic stem cells? Yeah. Let's go take a look at that. So this room, we basically is dedicated for studying human embryonic stem cells. We really don't use this room for anything else because these are special cells. They actually need to be taken care of every single day. So
That's your job?
Yeah, we're very sensitive to cell death and easily get infected. So every time you open or touch them, you need to just clean your hands.
Thomas was asking if it's your job to take care of it.
Oh yeah, I do take care of it sometimes when Richa is not here.
Yeah,
and I have separate experiments for it. Perfect. Yeah, so these are a cluster of human embryonic stem cells over here. There are actually hundreds of cells over here. This is what they look like when they are undifferentiated. But if you treat these cells with certain conditions and factors, then each one of these cells will actually start differentiating into whatever protocol you want. We've been able to differentiate them into neurons. It takes actually a month or 2 months, so it's a long, tedious process. We've also been able to differentiate them into cardiomyocytes, and these are cells that form the heart. What's amazing is that when they go into forming cardiomyocytes, you can actually see them beating in a dish. It's the wildest thing I've ever seen.
That sounds creepy.
It does. Tells you you're alive. And so they are very powerful cells. If you want to know how does a process change as one cell becomes another, and that's exactly what Sahil studies and what Reacher studies do. Awesome, wonderful. Awesome. Thanks, Sahil. Yeah.
All right, I'm gonna go ahead and thank you now because I'm. But Dr. Deshmukh, thank you so much for showing us your lab. This was really fascinating, and I think that the students, as they watch this, are really gonna love what they see. So we really appreciate your time and as well as your groups. Thank you so much.
Absolutely. Thank you for coming, and we'd love to see the students. Give them a tour in person.